Thanks, Nicole. And thanks, Nigel, for those kind words. I'm afraid I'm one of the dinosaurs. Exactly 1 year ago next week, our Yeti was installed in our lab. And in fact, I looked it up this morning on our calendar, and our training session was on Thursday. So next week, Thursday, 1 week from today. So it's really nice that we're meeting today, exactly 1 year after ours was installed. So just a little bit about who we are. We actually are primarily sponsored by the Cancer Center. We also are sponsored by a Skin Diseases Research Grant. Our instruments are probably a little unusual for most core labs in that one prime manufacturer is missing. We have two Beckman Coulter Galii, as I like to call them. We have a Cyan. We have a Yeti. We have 3 sorters, 2 MoFlow XDPs. One of those XDPs was the first XDP ever installed. And our MoFlow, our other XDP was a legacy, number 24, that was upgraded by Propel to be an XTP, so it's still functioning very nicely. And we have a Helios mass cytometer. Our customer base is fairly large. We're probably the biggest core on campus. We have 182 different labs that used us in the past year, 341 different investigators. And over the last year, we've actually done work for 44 different investigators on the Yeti. And as you can see in the graph, it started out rather slow. We really didn't let anyone on it or let anyone drive it until we were comfortable ourselves with the software, and also until the software was a little bit more developed, so we felt people wouldn't have a tainted experience. But as you can see, the last 2 months have really, really expanded. And I would say the Yeti's every bit as busy as our Galileo's instruments are. Just on another note, one of the reasons we purchased it is we have had a long-standing relationship with Propel, originally with Cytometry. I actually had one of those, was it called a Cicero unit? And the Cicero unit worked really well, and those guys went on to form Cytometry, and so I had some trust and confidence in them because that little piece of, it was an add-on to an older cell sorter which actually doubled its sort speed from 2,500 cells per second maximum to 5,000, which was incredible. It did everything they said it would, and that started off a really good relationship, which also I think was somewhat based on the fact that we're really close to the Denver airport, and TDAR like to bring customers through on their way back to the airport. So, number 1, we're going to go over a few things that we've learned or looked at about the instrument's performance, and then just a few application notes. So we got this instrument a year ago, and it had 28 different color parameters. We have a fully loaded baked potato, 5 lasers, and oh my gosh, how do you handle 28 different colors? Where do you start? So the first thing we did was try to determine what type of voltage settings should we use? Where's the appropriate spot that we're actually going to be able to tell the difference between negative and positive. You don't want to go too low because you're not going to be able to see anything. So the first thing we did was we just took 8 peak rainbow beads from Spherotech, ran them on all the detectors at a series of different voltages from 400 through 900, something like that, and looked at the CV of the 8 peak, one of the beads that we could actually see the best across all of those voltages. And then we plotted the CV by the voltage, and that point where the CV dropped was the point that we thought you shouldn't go below. So that was our standard, our starting voltage for our customers. We made this great chart that was laying by the instrument so people could see, oh my gosh, where I set it. The software was a little rudimentary when we first got it, so there was no way for us to set those voltages up to begin with. It was each time you had to go in and set the voltage. And so we just had the chart by the instrument and said, let's start here. And you'll notice the red circle kind of circles the order that we had the chart in. And that was basically by laser and wavelength, and then the wavelength of the floors. But as you know, it really, the fluorescence labels aren't in that order, and the order that you choose the voltages aren't in that order, so it was a little confusing. But we went with that, and then I would say over the course of the next 6 to 8 months, maybe even 9 months, we kind of worked with those voltages, and we'd find some were like really off, that just didn't work. We'd make a little note, scribble it down, we'd kind of look at the compensation as we did things for people, and we just kind of flew by the seat of our pants. Because that was the best we could do. But we got it working pretty well. And we had a lot of people walking in the door that saw all of those colors and really walked in the door with 14 color experiments that they unfortunately really didn't plan too well. So we thought we had a pretty good idea, but we thought there must be a better way that we can, let's really firm up this exploration of where we should be. So we worked with this paper from Mario Roederer's group. This was the first one published in, I think, 2008 or 6. He has another one, an update from 2012, where he talked about finding the sweet spot. Where's the sweet spot? Where you're really going to get the best signal to noise on that parameter from your instrument. And the advantages of using this sweet spot theory is that you're going to reduce the compensation because you're not going to have too high of a signal in the secondary detectors. So your compensation is going to be less than 100%. That's going to reduce the spreading error. And in turn, that's going to increase the dynamic range of your experiment. So originally in his paper, he used 8 peak rainbow beads. And then in the second paper, they switched to cytocal beads, which are from Thermo Fisher. Those are the original Duke scientific beads. We actually found these to be a lot easier to use also. So you can see the plots here, the 8 peak rainbow beads on the left, the cytocal beads on the right. These are concatenated files through FlowJo, so that you can see the various different voltages. So on the X, each sample was run at 400, 500, 600. And if you notice, does this work as a pointer? Maybe not. The ones on the left, it's really hard to follow which bead is which bead through those plots visually. Visually, the ones on the right, it's because there's only 6 and they just fit on the scale a little better. I really liked using them. They were just much easier to work with. Also, the dyes that they have incorporated, hard dyed into the beads, are more similar to what we actually use in flow cytometry. So we use these beads. And then this is how we actually did the measurements. Linearity is defined as the mean fluorescence intensity of bead M2 minus the mean of bead M1 divided by M1, the mean. Signal to noise would be the mean intensity of M1 divided by the background. And the background was actually determined by using the negative beads from BD comp beads. I use that because that's what Mario did. And also, in the violet channels, we didn't use those because they have so much background. But he just felt that that was a better negative. I found that pretty much they were similar to the lowest bead in the cytocal beads. And as you can see, this is an overlay of the beads in the APC channel. And the negatives actually lined up really well. So the bottom graph shows our sweet spot being the point where the signal-to-noise data peaks. So we did that for all 28 channels with cytocal beads. This is a comparison of our APC channel data to the data for APC channel on an LSR2 in the first paper. And you can see it looks pretty comparable. Our CVs never really flattened out quite as much as theirs do. And you can also see that pretty much the linearity and the CV kind of follow each other pretty closely, which is why we originally used CV. We knew that from this published work originally. Also, if you compare the look of this data on an LSR2 versus a Yeti, you really notice that the noise on the LSR2 just never really pulls over like the Yeti does. So it's just a difference in instruments, but it didn't seem to, the method still seemed to work pretty well for us. So here's a few of the Yeti sweet spots. The x-axis are labeled, so if it's B, that's the blue laser, and then the filter combination. So these are, 1 red and 3 blues. And so we did all of that and just plotted the, found the sweet spots, made our list. UV didn't work very well. UV didn't work at all. I think the dyes just aren't excited by the UV. There was one that there really was no excitation at all. The others just kind of marched up the scale with really no difference between them. So I tried some other beads. I tried the Spherotech Ultra Rainbow beads that are better for other like red and the far red channels. They didn't work for UV either. So we're still going with our original seat of the pants method. So here's our new chart for our customers. We changed the order so that the order of the fluorescence is now the order that you're going to choose them on the instrument in the software. And that was done because until recently we really still couldn't set our voltages. But our current procedure, and I think maybe this has been changed now with the latest software upgrade, is we have a run list that has these voltages in, and we have people, load that run list that sets the voltages, then go back, start over, and make your own plots. But I think there's a template that you can put in now. Is that correct? Okay. But that's within the last 2 weeks for us. So the next one is acquisition speed. And by this I mean how fast can you collect data without losing data in the electronics? How fast can the electronics go? And this one's really for you, Dan Fox. Because when we were talking about purchasing the Yeti, I asked Dan Fox, how fast can it go? And he said, how fast do you need it to go? I said, I don't know. How fast can it go? And he goes, infinity? So I thought I'd check him out. It's a racehorse. This is how we did it. We took Bang's beads, dragon beads. They're 1 micron, really bright green beads. And we made an initial dilution because they're so concentrated. So I just kind of took 100 microliters into a mill. And then I made serial dilutions of that, 2-fold serial dilutions up to, I think, about 512 or something like that. Then I ran the highest dilution and assumed that that was 100%, that the instrument was going to be able to see all of those beads. And then we ran each of the increasing concentrations and looked to see where did the count fall off from the theoretical. It should have been 2-fold higher for each concentration. And here's what we got for a few instruments. The Gallios is in yellow. A guest cytometer is in orange. And our cyan is in green. So you can see that, and the dashed line is the theoretical, where they should be. None of these really go beyond about, what, 20,000 without falling off. I did this several years ago, actually, for like a caliber and an FC500. They really fall off at about 2,500. So these are actually doing fairly well, but none of them really meet that theoretical dashed line going up. There's the Yeti in blue. You can't see my dashed line anymore. It goes up to about, what is that, 105,000. It really would go beyond that. The problem is I start getting so many coincidence events that it's hard to really calculate the number that's there. As it is, I had to kind of make some gates for, okay, these are the doublets, obviously, and kind of like add those into the calculations. But I don't know, Dan, I think it might go to infinity. You might say, well, gee, is that really important? We don't really run that fast. Sometimes you do. Sometimes if you're doing microparticles, there's a lot there, or bacteria. Or if you're doing absolute counts on those types of samples, you really want to know that your instrument is really seeing everything that's there. So that Yeti's a racehorse. Next, we wanted to look at sensitivity a little, just do some comparisons. And I felt that the best comparison we had in the lab was probably our Gallios instruments. It's our newest analyzer. And they're known to be really good analyzers in terms of sensitivity, resolution, dynamic range. This is a slide that I borrowed from Ryan Dugan, where he compared what he calls instrument overlap ratio between a fax caliber, an Accuray C6, LSR2, and a Gallios. And you can see that the Gallios actually came out on top. And so I'm using this just to show that our Gallios is really a pretty good standard to measure things against. So then we took a series of different antibodies that were a kind gift from Sysmex and just looked at the signal-to-noise ratio on the Gallios versus the Yeti. And in particular, the Gallios that we used has a 561 laser on it, so we felt that the PEs were pretty comparable. And sorry, I'm having a hard time reading. Okay, the Yeti's in blue. Gallios with the 561 is in orange. And notice that the Y scale is on a log scale, just so I could fit everything on. And the individual values are in the table on the right. The Yeti actually outperformed the Gallios on almost everything, except the PAC blue and the Pacific orange. And the APC was slightly lower. But I was amazed, I truly was amazed that it was not only equivalent, but in some cases tremendously higher. If you look at the PAC blue and the PAC orange, now the PAC orange, everybody knows, I mean, that's the dimmest candle in our box. And the Gallios was higher, but the filter range on the Yeti is much lower. So it was amazing that it compared as well as it did. The APC channel, where they were, the Gallios was a little bit higher. Filter range is kind of equivalent, but it wasn't that much different. So now we wanted to look at absolute counts. Now the software wasn't quite working for absolute counts yet, and I mean as far as actually giving you the calculated absolute count, but about, I don't know, 3 or 4 months ago we actually had the volume keyword added to the file, so we were able to calculate absolute count, but we wanted to know how accurate is the absolute count? How precise is the absolute count? Just as a matter of review on the Yeti, on the acquisition modes, high throughput has multiple samples in the line and there's no boost in the pump, and that's what they really recommend for using for absolute counts. In non-high throughput, you're going to have one sample in the line, and the pump is going to boost it for 5 microliters and then slow down to the program speed. So as way of background, we had this theoretical controversy in the lab. And I said, no, you have to do that. If you're doing absolute counts, you have to do high throughput. And Christine said, no, I think if you just kind of wait for it to settle down a bit and then tell it to record, you're going to be probably equivalent. So we thought, okay, we're going to test 3 modes. We're going to try high throughput mode. We're going to try acquisition mode that we pretty much knew wasn't going to work. And we're going to try Christine's method, which was acquisition mode, let it stabilize, on setup mode, let it stabilize, and then record. We took some Beckman-Coulter flow count beads. So those are the beads that you spike into samples for doing absolute counts by volume ratios. And the assayed value was 989 per microliter. So I made a 1 to 10 dilution of that. And then I ran that same dilution 10 times on the 3 different methods of acquisition. I set stop on volume for 30 microliters. And the flow rate was 1 microliter per second. And we calculated it based on the number of beads that we found divided by the volume keyword. Thought, okay, it's in the, that should be correct. And here's the data that we got. So the high throughput mode was really precise, really low standard deviation. I think the CV on the thing was 1% or something. Acquisition mode was terrible. We knew that was going to be awful. Setup mode, that didn't really look too good either. And the count per microliter was kind of all over the place, but none of them were at 99, which we should have had. So I talked to Mike Kisner about it, and he said, we're looking at that volume keyword, and we're just not really sure that it's really recording correctly. Try just using 30 seconds, which, so we did that, calculated using just 30 seconds. And things actually looked a little better for both high throughput mode and Christine's method, and still pretty bad for acquisition mode. So the very precise measurements, but none of them were really 99, which they should have been. We checked the counts on the beads with a Vicel. Now our Vicel is, it's meant to be a particle counter. It's under service contract. Ran the calibration beads on it. It was right on. And it recorded our flow count beads as 101 per microliter. So that, I took that to be pretty correct. We had some other beads, so I tried the same thing with some really old FlowCheck 675, but I counted them on the Vicel, and we got a lower count too. So currently, as it stands, it's really a precise counter in our hands, but it's not terribly accurate. But I know I talked to Mike, and he said they're kind of working on exactly how it's measuring the volume and looking at maybe some different ways to do that. So now just a few little things about some application notes. Microparticles. We've done a little microparticle work. I don't pretend to be an expert on microparticles, but I wanted to take a look at, how does it compare? So I actually found this paper by Bob Zucker on microparticles, and this plot on the right is microparticles on a Stratodyne S1000, which actually was optimized specifically for microparticles, and they didn't really say how they optimized it because there were some patents pending on it. But he had a plot of polystyrene beads with the lowest being 200 nanometers on up to 1,000. And we ran some bangs, submicron beads on the Yeti. So those are at 200, 500, and 800 nanometers. And I thought they really looked pretty equivalent. We actually also had the bangs nanoparticle beads, which are 50 nanometers and 100 nanometers. I couldn't resolve them on the Yeti. So I think it's a good microparticle instrument. I don't think it's blowing any of the other good microparticle instruments out of the water. But on the other hand, they didn't really design it to be a microparticle analyzer. It's just looking good. Interestingly, if you look at the 488 forward scatter versus the 405 forward scatter, you'll notice that the 405 has a lot tighter peak. The CV is a lot tighter. which is really helpful for microparticles. Christine had noticed that, sometimes people bring samples in and they've had a poor lyse or not lysed at all, and red cells just really get in the way. So we spiked some red cells into a lysed sample of human blood, and the plot on the left is the Gallios, and we color evented on CD3, so you can see the lymphocyte T cell population in red, and on the Gallios, the red cells clearly just overwhelm it. There's no way you could find that without being able to gate on a specific marker. The forward scatter on the 48 laser on the Yeti, pretty much the same thing. The red cells kind of overwhelm it. But if you look at the 405, you can see that little red population is really distinct. So you can use that to, find your lymphocytes pretty clearly, even if the sample has a suboptimal lyse. So we actually do this quite a bit and find that 405 forward scatter is really helpful. This is one of the first samples that we ran on the Yeti. So we do a lot of side population. If you're not familiar with that, it's a stem cell population, very rare event, based on the efflux of Herx 33342. That assay was originally developed in the Margaret Goodell lab on a Fax Vantage. And then they moved on and used a MoFlow, and pretty much the MoFlow and Fax Vantage have been considered to be kind of the gold standard for that assay as far as resolving the fluorescence of the Herx in the red and the blue channels. So we've always done side population on our XTP instrument, which is our only sorter with a UV laser. But since our Yeti has a UV laser, we had this sample just a couple weeks after the Yeti was installed, and we had a little left over, so we said, let's look at it on the Yeti, and it was, oh my gosh, that's fantastic. I don't know of another analyzer that can really clearly give you that distinct side population. And the top left plot on the MoFlow was the verapamil standard, so that's a negative control. It blocks the efflux. And then the plot on the left bottom was the XDP, so it was 1.65%. And we ran it on the Yeti and got 1.68%. And the other thing I should point out is this was a cell line. This is a melanoma cell line. So it looks a little bit broad on the XDP. Maybe the XDP was having a bit of a bad day. I mean, we certainly tried to align it, but the alignment, I mean, the G1 population, which is that big population in the center, actually looks tighter also on the Yeti. So I think it's just a matter of, the XTP is a jet and air sorter, and the Yeti has the advantage of a cuvette thing. So in summary, the pros for us is we really actually think the software is really intuitive, still under development. It's come a long, long ways from when we first got it. That whole driving aspect of it, by that I mean the acquisition, go, pause, restart, really wasn't quite there when we got it. And so one of the advantages of being number one is you get to kind of make your wish list, and it happens fairly quickly. So we kind of designed that, so if you have any complaints, don't see me. We think it's a really flexible instrument. I mean, 5 lasers, 28 colors. It can do amazing things. We think it's really fast. It is infinity. I think the light scatter is a work of art. If you put mouse bone marrow on there, you see populations that you really can't see on other instruments. They look like kind of little string of pearls. I really like the compensation, the automated compensation. I think it's really easy, and it seems to be working really well. I have compared it to Kaluza and Flojo, and if I try to gate tightly in the same way, I pretty much get the same compensation. I also really like the fact that it's quiet. I've spent over 30 years in a flow lab with noisy pumps and fans, and that thing's quiet. It's relaxing. Our current con is the software bugs. We're kind of crashing a lot and telling people not to go exploring because that's a great way to crash it. Although I think the latest version is improved. I managed to recover from a run list error the other day by just kind of waiting. So that was great because in the past we had to just kind of start over. I mean, completely start over. And if you're running, a 96 well plate and you crash on number 85, you don't really want to start over. And a big thanks to Christine Childs, who works with me, and she's really been kind of the lead person on the Yeti. And then the whole Propel team, Mike, Reed, Mark, and Eric, who have really been on top of things, coming down with some initial hardware issues. The fact that we're only an hour away was really a good thing, and also just online support, etc. So, questions? Yes. Just on one of them. No. We've actually been pretty happy with the Galios 561. We got it initially because we really needed an instrument that could do the red fluorescent proteins. And I was not happy with the fact that it has the yellow laser is collinear with the blue, and I thought it was going to be a big problem. But I was really surprised that when we compared it, that it really wasn't that big of a problem, and the compensation is still reduced. So if we're looking at FTSE out of PE without the yellow laser on, it's like 15% compensation. With the yellow laser on, same filters, same voltage settings, it's 5%. So it does reduce it, and no, we haven't really noticed that.
I was wondering, is it making, yeah, okay. I was wondering about the sweet spots that we were mentioning before. Do you think that they can be applicable to different instruments with the same settings, or it would be something that every single instrument should be running to find?
Theoretically, you're supposed to do it on each instrument because it can point out problems with PMTs and problems with filters. That paper, it's actually really complex, and the sweet spot is just one part of it. It's meant as a paper to show you how to standardize within instruments within labs. So it goes on, it finds the sweet spot, and then it goes on to say how to standardize daily. And it actually uses comp beads, I think. After you find the sweet spot, then you use the comp beads with your actual antibodies to really verify the sweet spot by making really slight changes to the voltage. Then you go on and standardize between days. And so I really think it still would be, you could probably take my chart and at least give it a try. And maybe not have to go from 300 to 900. They went in increments of 50 in the paper, and then they said when they found the sweet spot, then they did increments of 25 around that. We didn't do that. I looked at the plots, and most of them I could see the peak pretty clearly. If it looked like there was kind of a flat spot instead of a peak, we went 25 lower or 25 higher. We just wanted a good spot for our customers to start. And, we do make slight adjustments from that, but it's a really good place to begin.
I don't know if this already exists because I don't quite have my Yeti yet, but are there plans to have that voltage optimization automated?
Yes, I could talk. I don't think we have any plans today to do it automated. Certainly it seems like something you did a study like this and you want the starting voltages. That seems like a really easy thing we could implement and just allow you to set default voltages for your system, and that way you don't have to load. I mean, that seems pretty simple and straightforward.
Yeah.
Automating this, Could we do it in the future? Yes. Do we have plans today? No.
There are actually people that have used like a pulsed LED laser in the system, and we initially thought maybe we could use the Yeti eye for that. And we kind of tried it, but it just didn't really seem to work.
There's some things you can use the eye for, and that's one of the things we've been talking about. It can help you system to system to help normalize. The biggest variation that you get in trying to go from one system to the next is the difference in gain in the PMTs. So PMTs are just pretty variable from one to the next. And if you take what would be pretty close settings that somebody has already done on this, went to your system, give you a good starting point. But if we could somehow use the eye to help normalize those settings from Karen's system to your system, it would take a lot of that variability out. So that's kind of one of those things we're looking forward to, kind of on the future of features that we can start bringing and ways we can use the eye to do more and more than it's just doing now. So there's some things we're thinking towards in that range.
Anybody else? Yep.
This is actually for both Dan and for Karen. The PMT is obviously the most variable. They're analog devices, and we're converting them. ADC background, I know every analog to digital converter that I've seen has a different fingerprint on where it's noisy or where it's not, and they're usually integrated in sets of 8 or sets of 4 on a single circuit card. So there's no standardization or itemization of a ADC. You have a wonderful thing in the ability to look at threshold and background on the threshold. If you had one more window that was apart from the threshold parameter, that you could actually see where the ADC background and noise factors were on a fluorescent parameter rather than forward scatter or whatever your trigger parameter was. You could actually see the distance between what we know is negative and what the ADC background would creep up and come close to it, and whether there was a defined gap between that. I think that view of background on threshold is incredible. I could see that as a tool even further for just one more window that you could toggle whatever fluorescent parameter you were setting. Because is this true in your case that ADC background is variable per ADC?
For the one it's less variable than it used to be, which is good. The variability that we've been seeing so far is pretty small. The biggest variable that you can get is if you have any grounding issues in any of the connections, any of the cabling, and that goes back to the days of taking your MoFlow and going through and rotating the connections one by one. So there are things we do to kind of look towards what you're talking about. So that'd be an interesting thing to look at and to see what we could automate or see what views we could include in the software. I think that's an interesting one to look at, yeah.
Karen, I have just, it's almost more of a comment maybe for later in the day, and that is, as regards absolute counting, you never really got where you wanted to be. And I got users that really want to be very accurate in that, and they actually want to count bacteria, okay?
Yep.
And I don't know what the Propel people have going on, but that's something maybe we can talk about later today, about how you're going to address the inaccuracy in absolute counts, because that's something I would really like to have more accurate, because I've found basically the same thing you've found.
Does anyone else have a system that does absolute counts, and have they checked it?
So internally, I just got a couple of new systems, and we're running the latest software. And in our initial tests, we've been running at about 5%, running about consistently about 5% below what the expected count should be. But our CVs have been really good, and it's been less than 10%. It looks much better than what you have shown there. So I think whatever's going on is getting better. I know they're looking at a lot of different things. There are some pump calibration things that can be done in terms of making sure the correct volume's being delivered. But at least internally in some of the trainings that we've been doing with our FAS team, the last runs that we did, our absolute counts were looking much better. So
What are you using as a standard?
it was from eBio. I can't remember I can look it up in a few minutes. And it's very similar to the Beckman-Coulter product. So
Okay.
it was a 1,013 per ml.
Okay.
So, or per microliter, sorry.
I guess I'll share our experiences with absolute counts because we did some comparisons. We have max quants in our core, and they're supposed to do they're a volumetric based flow cytometer too. And we saw it's funny, you had like 69% of the beads that you got on your Yeti. Our Yeti was exactly the same, 69%. And that was actually in acquisition mode because I didn't appreciate the pump cycling that you spoke of. But we're seeing a kind of similar lower level of beads than we'd expect. And this is with the Spherotech beads, counting beads you spike in, they're FTSE labeled. We've also looked at CD checks, which are stabilized blood product that you're supposed to get they give you an actual value of CD45 cells that you're supposed to pull out. And we've also seen that those are lower than what we expect. But our max quant never, with those CD checks, it never quite gets to the perfect level that they recommend either. So it's kind of, I think it's a hard problem for all manufacturers.
Just throw a couple comments on that real quick. We're kind of going through right now and doing an audit through all the different areas because there's kind of the setup and the operation of the system to make sure we're getting the most consistent way of setting it up, getting things ready, getting ready to record the data right, an audit on the whole data trail like we talked about at one point. The volume that was being recorded wasn't quite the right volume, a couple of those pieces. So making sure we take the information we have and do record it, and looking at kind of the intrinsic performance of the pump itself at different speeds, at different flow amounts. So there's a lot of different pieces that are going on. And so like Matt was talking about, we've made some improvements and we'll continue to keep doing that. It's something we're looking at. It's definitely a high priority. And we've kind of, as we've been working through some of the other priorities, this one has now bubbled up and has a lot of effort going on with it right now.
So the small particle got down to 200 nanometer. Is that correct?
Yeah, 200.
So can that be optimized at all? I think it's advertised at 100 nanometer for the Yeti.
Less than 0.3 or 300 nanometers. The inline sheet filter is 0.22 microns, so that's kind of where your borders.
And ours is point.
You guys have a 0.1, right?
Yeah, a 0.05. Yeah.
Okay.
We'll have our optical engineer here in the afternoon session for hardware, so we can talk more specifically about that.
Okay.
He can answer the questions about the small particle stuff much better than anybody else did.
Okay.
Anybody else? No? All right. Thank you, Karen.